Hi, everyone. Thanks for coming. It's a pleasure to be here today with the group. On behalf of OraSure and DNA Genotek, we're pleased to participate in this session titled Molecular Diagnosis with NGS for Neurological Infectious Disease Conditions, featuring Dr. Frank Middleton and Dr. Forrest Wright. My name is Scott Rabuka. I'm with DNA Genotek, a subsidiary of OraSure Technologies, and my title is Senior Director of Molecular Products. Within OraSure, we have three main lines of business. One is sampling tools or collection products. The next is microbiome services and analytics. And the third is diagnostics and testing applications. The focus today will really be from the sample collection side of our business. Within OraSure, two subsidiaries, DNA Genotek and Novosanis, have a broad portfolio of collection products focused on application areas of human genomics, microbiome and metabolomics, infectious disease, and oncology. Where necessary, we have gone down regulated pathways for 510(k) clearance and de novo applications, as well as CE-IVD markings of a number of our products. So today, the product of interest for the next two talks is the OraCollect RNA product. It's a product that uses a collection sponge from an oral sample collecting saliva and buccal epithelial cells. It's connected to a double-ended cap. On the other side of the cap is a stabilizing reagent for stabilizing the RNA at room temperature for 30 to 60 days. So that's really the product that's enabling the work of the next two speakers. So I'd like to introduce our speakers. So Dr. Frank Middleton is a professor in the Department of Neuroscience and Physiology, Biochemistry, Psychiatry, and Pediatrics at SUNY Upstate Medical University. He's the founder and director of the SUNY Molecular Analysis Core Facility, which specializes in next-gen sequencing analysis. His research pursuits using high-throughput genetic, epigenetic, genomic, and metagenomic approaches have led to scientific discoveries about the role RNA plays in the human body, including the early diagnosis of autism spectrum disorder, Parkinson's disease, and mild traumatic brain injury. Dr. Middleton's research has revealed that particular RNA and microRNA related to these applications were made in brain cells and found in saliva. This is one of the key reasons why saliva is providing a window into brain function. Dr. Forrest Wright is Director of Assay Development at Quadrant Biosciences. He earned his Ph.D. from Upstate Medical University in 2017 and has managed multiple research and development teams operating across the fields of cellular and molecular biology, as well as preclinical pharmacology. In his role at Quadrant, Dr. Wright leads his team in the development of novel saliva-based assays in the fields of genetics, epigenetics, virology, with a primary focus on neurodevelopmental and neurodegenerative diseases and disorders. These assays will be revolutionary in the advancement of diagnostics that will aid in the early detection of global health priorities, such as, again, autism, concussion, and Parkinson's disease. They rely on at-home saliva-based sample collection, ideal for telehealth-enabled practices that allow for increased access to much-needed patient testing. So with that, I'll pass the podium over to Dr. Middleton. Thank you.
Thank you very much, Scott. And it's a pleasure for me to have the opportunity to tell this group about our recent success over the past decade in harnessing the power of saliva to learn about brain disorders and infectious diseases. Spit is where it's at, okay? And it all starts with saliva, in case you didn't know that. If you've heard about different diseases where there's changes in the gut-brain axis, the beginning of the gut, obviously, is the mouth. And one of the things I'll try to convince you is that we can learn a lot about these conditions by really starting at the beginning. In addition to embracing saliva as a biofluid for discovery, we also embrace AI and machine learning algorithms because we take the approach that we don't know the answer before we actually examine the data. And that's been very fruitful. The goal of the research that we've been engaged in is to develop the most sensitive and specific biomarkers for early detection of brain-related disorders and conditions, as well as infectious diseases. Because earlier you can detect it, the earlier intervention can occur, and the better the outcomes can be tailored to those individuals. Today I'll focus on describing our work in autism, Parkinson's, and COVID-19, but we've obviously had other things stewing as well, including work on concussion, TBI, and many other conditions. So let me begin with a discussion on autism. So autism, as many of you would be familiar with, typically presents at or around age 3 with behaviors that indicate changes in social interaction, social communication, and frequently restricted and repetitive behaviors will emerge. The incidence of autism has been increasing over the past few decades and presently is approaching 2%, approximately 1 in 44 children, although the rates are 4 times higher in males than females. And these rates occur across races and ethnicities, although access to the early evaluation and diagnosis of autism is something that exhibits a great deal of disparity among different races and ethnicities. So one of the things that we wanted to do was develop a method of detecting autism at the earliest possible age, because the current wait time for these evaluations is 9 to 12 months in the US alone. And that's only in populations where there is ready access. Other disadvantaged populations have to wait twice as long frequently to be evaluated. And that means a delay in the intervention, a delay in helping those children develop the best that they can. So using saliva, we can overcome many of the bottlenecks because we can gain insight potentially into what's going on to a level that's sufficient to assist a clinician with making a diagnosis of autism earlier. You might think that we would want to study genes in saliva at the level of the DNA, but studies of autism looking for candidate genes have only uncovered clues that explain potentially 10% of the cases of autism, leaving the vast majority unexplained. So we thought perhaps we could gain new insight into potential causes or contributions to the condition of autism by looking for epigenetic factors that relate to how the genome is expressed. Where did we look for answers? We focused on 2 molecular classes of RNA that you find in saliva at very high levels, microRNA and Pee-wee interacting RNA, or piRNA. Both of these share not only their length, they're approximately 20 to 28 nucleotides in length on average, but they also share the fact that they suppress gene expression. MicroRNAs act to suppress mRNA stability or protein translation, and piRNAs will suppress repetitive elements in the genome. When there are changes in the level of these, you have consequently alterations in cellular expression of proteins or repetitive element genes. And this can have very strong effects on the cells and the tissues that they're found in. One important aspect about these non-coding RNA classes, it's important to recognize that they don't stay in the cells where they're actually synthesized, but they are transported readily in biofluids, frequently in exosomes or microvesicles, where they can travel throughout the body. So it's conceivable that we could detect in saliva microRNAs that have a different origin, potentially an origin in the brain. And if so, that may be particularly insightful for studies related to autism and other neurological conditions. So why would saliva RNA provide insight about the brain? It all comes down to connectivity. There are 5 pairs of cranial nerves that innervate the mouth. You use these nerves every time you try to speak, swallow, salivate, make a smile. All of the activity of those nerves depends on the transport of molecules, including RNA, proteins, and neurotransmitters down these cranial nerve paths. These nerve paths are not new. They're the same nerve paths that the RNA virus rabies uses in order to infect the salivary glands when an individual person or animal happens to have been bit. So these pathways are well worn. They provide the opportunity for RNA to be deposited in the salivary glands as a result of brain activity. To gain further insight into what the saliva may hold related to brain, I can actually also tell you that using shotgun mass spec sequencing, we're able to identify more than 30 ASD candidate genes present in saliva at the level of protein, and more than 100 proteins that have been mapped to proteomic networks involved in early brain development, like the first two of the first three that you see on that table on the right. This indicates that saliva is doing much more than just acting as a fluid for digesting your food, but in fact appears to provide a readout into brain function. Now, why would saliva provide information about the immune system? It turns out the oral cavity, as many of you may know, is a primary defense against infectious agents. It is a site where there are a lot of antibodies that are made in the cells that surround the salivary glands, which you can see in the lower right of this image. IgAs and IgGs made in plasma cells are released in the saliva, and we can measure those, which comprise about 10% of all the proteins in saliva, as well as other proteins involved in defense against infectious agents. It turns out that the microbiome, as I mentioned right at the beginning, is altered in ASD, as well as Parkinson's disease and other neurological conditions. So by studying saliva, we have the opportunity to measure not only brain-related molecules that appear there, but also molecules very much involved in immune response in the gut-brain axis. So using next-gen sequencing, learning everything that we can about what's present there and how that's changed in autism, we're able to feed the data into machine learning molecules and develop a classifier that was over 85% AUC in the training, cross-validation, and the naive holdout set of children who either had autism spectrum disorder, non-autistic developmental delay, or were neurotypical. And you can see the performance curve. It looks fairly impressive on the right side of this. The composition of this classifier was approximately half human and half microbial, supporting the utility of using that readout in saliva that comes from the gut-brain axis. The sensitivity and specificity of this, when we compare it to behavioral-based diagnostic measures for ASD, actually holds up quite well. The positive predictive value of the saliva-based NGS classifier was 91%, and that's much higher than you see with the gold standard Autism Diagnostic Observational Scale, which is listed right below it in this table, which only has a positive predictive value of 22%, where that behavioral-based clinical gold standard struggles is in differentiating non-ASD developmental delay from ASD developmental delay. But that's exactly what we trained our model on. We included the most difficult classifying task in what we developed in the classifier. The classifier performance is very similar to what you actually see for infectious agents, such as rapid strep tests and flu tests. And it's certainly better than the PSA test. So autism was successfully addressed, and we thought we gained some new insights from examining saliva. What about Parkinson's? Just one slide here. In a similar manner, we collected saliva from very well clinically characterized subjects with Parkinson's disease, and we examined changes in the expression of the human non-coding RNA as well as the microbial content that's there. Now, the oral cavity is particularly interesting in Parkinson's disease because we know that early in the disease, there are changes in the way individuals with Parkinson's swallow, later on in how they speak, and the neuropathology of Parkinson's includes these classic Lewy body protein inclusions, which you find in the salivary glands of subjects with Parkinson's, not just their brain cells. So the disease is there, it's in the mouth, and it affects what we are actually getting readout on. The performance of the model we were able to develop in Parkinson's even exceeded that which we achieved in autism spectrum disorder, consisting of several microbial features and non-coding RNA features, but the sample size is much smaller than what I showed you for the ASD studies. So this is currently undergoing a larger validation study, but we're very excited about the prospects. Finally, the third story. We've lived through a pandemic, right? We've all been reminded of that as we enter this display area here at this convention, the conference. Early in the course of the pandemic, there was clearly a need to have more tests capable of being performed, and ideally less invasive, that didn't require deep nasal swabs. So we responded to this need by developing a real-time qPCR test that was as sensitive and specific as we could make it, that was as resistant to mutations as we could make it. And we certainly didn't just adopt the CDC primers and probe sets. We also made use of the swabs that you heard Scott mention at the very beginning, because they not only stabilize the RNA, but they also inactivate the virus and many other viruses so that we could use the fluids in pool testing development without exposing the lab workers to hazardous potential threats. So we validated the sensitivity. We went through the whole steps and procedure to receive FDA emergency use authorization. While we were doing that on the individual tests, we developed a pool test methodology so we could combine up to 12 samples with no loss in sensitivity and rapidly process in a single lab more than 30,000 samples a day, which we did during the peaks of the pandemic. The best-in-class designation was achieved for the saliva testing individual and pool test methodology that we developed. Now, because of the stabilization of the RNA, we're also able, with the same sample, the same RNA that we've purified for qPCR, to subject that to whole genome sequencing to monitor the emergence of variants in the virus. And from the same RNA sample, we also looked at host responses to see if there were differences as different lineages came up, and differences related to subject characteristics, like their age and their sex and their vaccination status. This graphic illustrates where all the positive samples that we actually sequenced were identified throughout New York State. That was our catchment area, and you can see we had more than 40 counties sending in samples. Our lab is located right in the middle, where it's the darkest, so we had more samples there than from other counties, but clearly we sampled broadly across the state. This is actually a video that's not going to play right now, but it would illustrate the relative abundance of the different lineages across time throughout the course of the pandemic. So all told, we tested more than 3 million saliva samples. We sequenced more than 31,000 positive, and we're still doing that work. We did RNA sequencing to determine unique patterns and shared patterns of host responses in more than 300 samples. And Quadrant has biobanked more than 100,000 additional samples to date. One of the things we've learned from the sequencing is that this virus is continuing to change. This plot shows the amount of mutations that are present with the different lineages. The different lineages are in different colors, and time is on the x-axis. And clearly, there were different periods of rapid mutation compared to others with the different lineages. We didn't expect this, but this stealth nature of the virus reminded me, if you think back to Jurassic Park, of the same stealthy way those dinosaurs figured out they really could take over the island and outsmart the scientists. just as the warning Jeff Goldblum's character delivered, that life finds a way. In fact, one of the things that this virus has obviously done, in addition to increasing its virulence, is increasing the load. Using exactly the same qPCR methods across time, we can now share with you that as this virus has mutated, we're detecting higher and higher levels. So lower CT values in the PCR test are necessary across time. This is perfectly adapting to us. We've also been able to see in the host RNA responses some very specific changes related to individual lineages, which I won't go into, but we can detect conserved large-scale changes that indicate in the saliva we have a fantastic readout of processes involved in viral defense, suppression of viral genome replication, induction of interferon cascades, and we can even identify a set of genes that are infection-specific, that are not changed simply due to vaccination. And that's what's shown here, a panel of about a dozen genes that are absolutely flat, no change due to vaccination, shown in the orange, that show a robust induction 3- to 4-fold for each gene. In the infected individuals, that's the blue line across all the genes, and a slight reduction or attenuation of that response when it's a breakthrough, when someone was vaccinated. So it's these types of insights that come from appreciating all that you can learn from saliva. So to summarize, saliva-based analyses have revealed some very consistent changes in human and non-human RNA that can aid in early diagnosis of brain disorders and infections, the rapid identification, full genome sequencing of microbial and viral genomes, easily accomplished alongside the profiling of host responses when the RNA is well preserved. And this enables monitoring the emergence of variants of concern, as well as possibly identifying viral sequence alterations driving greater pathogenicity, greater virulence, and potentially host genetic factors involved in responding to that threat. So thank you very much. And I'll invite my collaborator, Dr. Wright, to join us on the podium and present his slides now.
Thank you, Frank. Yes, I'm Forrest Wright from Quadrant. I'm here today thrilled to have the opportunity to talk to you about the exciting things that we can do with your spit. More specifically here to talk to you about some biomarker discovery that we've done, I hope everybody took everything that Dr. Middleton just said as context for some of the products we're looking to develop. So we'll discover some biomarkers for neurological conditions as well as infections. Just a quick order of business at Quadrant. We work with our academic collaborators, such as Dr. Middleton, among others, provide us the expertise we need to establish and develop these clinical assays. We work to improve diagnostic accuracy and patient access through the research, development, and commercial implementation of these assays in both clinical and public health settings. So, as you just saw, all from saliva, right? We can take these saliva swabs, we can extract from them genomic DNA, large RNAs, small RNAs, such as microRNAs and piRNAs, as well as viral genomes. And using NGS as our central technology, but as well focusing on the kinds of sequencing we can do with traditional Sanger sequencing, and adapting many of those to qPCR settings as well, so they're a little bit easier to implement in the clinic. Really take saliva and just project that out to a number of different diagnostic tools. So just up front, taking a moment to assess NGS, particularly on small RNAs. In our development, we took a look at the bioinformatics options that were available to us and found many of them dissatisfying. So we took it upon ourselves as a small company to develop our own tools and really get to the heart of being able to use these tools for ourselves, develop something that we find more accurate. So the pipeline that we've developed for our bioinformatics is not just use a bow tie aligner, set it and forget it, take the data and run. We've developed something a little more accurate that allows us to develop the kind of high quality results with low variability that we expect from ourselves and to be able to provide a diagnostic product to our patients. I'll just take a moment to highlight a few of the products we have in development. Each of these is planned to be through regulatory by the end of this year. So 2 products for autism spectrum disorder detection, one Genotek, a whole exome sequencing based assay, and then 2 epigenetics assays, one for autism and one for traumatic brain injury or concussion. Again, those focused on small RNAs. And then taking the amazing tool we have in our particular COVID detection and then layering in a few other respiratory targets into a multiplexed assay. So in this case, these technologies allow us an opportunity to look at chronic diseases or disorders such as autism, brain injuries in the case of concussion, and acute infections for respiratory illnesses such as COVID. First up, talking about autism. In autism, as you just heard discussed, autism has a highly heterogeneous etiology, and genetic testing is really only able to account for about 10% of those cases. And there's not currently an epigenetic molecular diagnostic test available. One of the keys for our development and key interests was highlighted in that the timeline from first suspicion, when parents bring a concern to a pediatrician, to a final diagnosis, getting through all those appointments, the scheduling, the traveling to specialists, all the different testing, is upwards of a year, often more. And that really hinders the opportunity for therapeutic interventions. And it's well documented that the earlier those are established, the better and improved outcomes for children diagnosed with autism. This also gives us an opportunity to meet an essential public health need with a growing incidence of autism and the rates of diagnosis, particularly in boys, increasing continually over the last few decades. So switching gears to an injury model, many of you who have kids who play sports, you know that you may feel concerned every time they run into each other if they're playing football or lacrosse or hockey or anything. And any of you who may have had a concussion yourself or seen your kids with a concussion, that can be really scary. And concussions carry a high risk of long-term post-concussion symptoms, and currently there's no testing that identifies the risk for those long-term symptoms. Being a parent myself, I know that the turnaround time in getting results for anything with your kids is really critical. And we, again, as with the autism test, we want to shorten that turnaround time, get responses back to parents and patients as quickly as possible. So, looking at chronic disease, chronic disorder, and injury models, with our sequencing work, we hope to be able to provide rapid test results and reduce that turnaround time to patients. One other aspect of being able to use this RNA sequencing technology is being able to convert it not just from the sequencing results that we have into something that's a little more feasible, a little more price tolerable, depending on the test, to patients. For example, in viral infections, we don't necessarily have to do sequencing to diagnose that, right? So we've done extensive COVID testing in our region across New York State. You heard we were doing 10,000, or multiple tens of thousands of tests a day at the peak of the pandemic. Here we're doing the kinds of multiplexing development that many others are doing, but with our particular test combining COVID with other respiratory diseases. We're doing the current sensitivity, specificity, and cross-reactivity development you would expect. But alongside that, with our sequencing expertise, we can also monitor and detect seasonal variants as well as emergent variants of concern, particularly relevant to COVID. So these are the tests we have in development. As I said, we're approaching regulatory and being able to get through these out onto the market this year. And finally, I threw some names together about all of the people who are really responsible for all of this work. And I thank you for your time, and any questions you may have, we'll take them. Thank you.